In the context of Valle del Cauca, specifically at La Calamba Farm, located 600 meters from the Media Canoa toll booth, on flat land between the Cauca River and the foot of Loma at 950 meters above sea level, a study was conducted to assess the impact of CZF technology on the cultivation of yellow hybrid corn. This study, carried out with scientific rigor, aimed to objectively analyze the effects on agriculture of manipulating and structuring liquids and gases through controlled signals. In this case, CZF technology was applied to the water used for drip irrigation. The team of specialists began their work with a detailed survey of the terrain, taking precise measurements at the water source used for irrigation. At 10:30 a.m., oxygen values of 3 ppm and a pH of 5.91 were recorded, thus establishing a preliminary database. The plots were prepared with 2 passes of a harrow, and the question arose as to whether the quality of rainwater could influence the study results. After careful evaluation, it was determined that the impact of rainfall would not be significant on the final results, since both the trial and control plots would be equally exposed. In the trial protocol, the technology-treated plot would receive at least 2 irrigations with structured water per month, and a minimum of 1, depending on precipitation in the area. As the crop progressed, any changes in the plants were closely monitored. Observations were made objectively, maintaining a scientific approach at all times to analyze possible differences between the treated and control plots. 60,000 plants per hectare were planted, totaling 120,000 plants on 2 hectares of land. Of these, 30% received treatment with structured water, equivalent to 36,000 plants directly benefiting from the technology in question. On October 3, 2023, a drip irrigation system was installed, applying CZF technology to the water in one of the plots, 30% of the 2 hectares selected for the study, while the rest of the plot remained as a control. The first germination irrigation took place on October 3, ensuring that both plots received the same care, with the only difference being the treatment with structured water in the test plot. Additionally, some seeds were previously treated with structured water to reveal their germination vigor, a process carried out in germination trays. Due to continuous rainfall in the area since October 3, the next irrigation was not carried out until November 7. During this period, healthy development was observed in the test plot, with robust plants and intense green color compared to the control plot. Significant root differences were noted between both plots, with more pronounced root development in the treated plot. The agronomist in charge of the trial expressed satisfaction at observing these differences, highlighting that the darker color of the leaves in the treated plot indicates a higher chlorophyll concentration, essential for the photosynthesis process. This observation suggests a potential increase in yield in the treated plot at the end of harvest and reinforces the idea of the good health of the plants in both plots. Additionally, color is a clear indicator of plant health. On November 7, an exhaustive evaluation of leaf and root development was carried out in the crop field. 2 specimens of equal height, 1.95 meters, were selected to compare the effect of structured water treatment. Regarding the stems, it was found that those of the treated plants had a diameter of 40 mm at the root neck, while the untreated ones showed a diameter of 28 mm. Regarding leaf appearance, slight differences in green color were observed, more intense in the treated plants. Additionally, a silkier and more resistant leaf texture was noted in this group. In addition to fertigation applications, 2 edaphic fertilizations were carried out using Permaction mineral organic fertilizers from the Abanimos company. In total, 800 kilograms of mineral organic soil fertilizer were applied. As part of integrated pest management, a soft application for corn borer and dalbulus control was planned, with an infestation level of 5%. The products selected for this application are Catrino and Ngio. On November 28, during a field visit and observation process, irrigation with structured water was carried out, followed by evaluation of floral development, pollination, and fruit setting. Detailed observations were also made on entomology and the phytosanitary status of the crop. During these detailed observations, extensive discussion took place on the overall health of the plants. It was noted that the leaves are well projected upwards, allowing for adequate distribution in the high crop density. Encouragingly, it was estimated that the normal production of 1 ear per plant has been exceeded, with the presence of 2 ears per plant. This finding is highly promising and suggests significant yield in the harvest. During the irrigation day, the agronomist carefully observed the differences between the treated and control plots. Notable disparities were highlighted, especially in root development and the initial vigor of plants receiving structured water treatment. Additionally, a difference in leaf color was observed, being darker in the treated plants. It is interesting to note that as the experiment progressed, there was a trend for the plots to equalize, as if the untreated plot were following the example of the treated plot, acting as a mirror to imitate. During the research in the Yotoko field, preliminary tests were carried out to evaluate the performance of corn cultivation with and without the application of technology. These tests focused on the morphological analysis of cobs, specifically measuring the percentage of ovary diameter in relation to the total diameter, both in treated and untreated samples. Measurements were taken in 2 scenarios, 1 cob with 1 measurement and 2 cobs with 4 measurements each. The results obtained were compared between samples with and without treatment. In the measurements made, a significant difference was observed in ovary diameter in relation to total diameter between samples with and without treatment. While the percentage of cob in relation to total diameter was 63% in untreated samples, in treated samples this percentage decreased to 59%. On December 6, a visit to the test site was carried out, where measurements of water properties used in irrigation were taken. During this day, irrigation of the crop was also carried out. The engineer in charge of the trial provided a detailed explanation of the results of the measurements taken. It was highlighted that the measurement of available oxygen in the water, carried out 1 meter away from the treatment, showed an initial non-uniform reading. It was observed that the water showed higher activity, with a reading higher than normal, which stabilized approximately 1 hour later. One of the specialists emphasized the importance of maintaining an optimal level of oxygen saturation in the water, indicating that being above 66% at noon would be most desirable from a biological standpoint. Additionally, the pH of the water was mentioned, which was found to be excessively alkaline, marking between 7.8 and 8. This alkalinity is beneficial for plants, especially in acidic soils. The intensity of color observed in the leaves indicates optimal chloroplast activity, as well as excellent fruit setting. These measurements are crucial for understanding the state of the water used in irrigation and its impact on the health and development of plants in the test field. On January 21, a visit to the test field was carried out to observe the corn drying process in the field. The climatology during this period was characterized by summery weather, with sunny days, high temperatures, and no rainfall, allowing for a drying process without inconvenience. From January 22 to 30th, a second visit to the field was made where the corn drying point was approved and manual harvesting followed by machine shelling was carried out. Despite adverse weather conditions, with strong winds, no plant overturning was recorded, indicating excellent root quality. After harvesting, the results obtained were evaluated. In terms of quantity, a total of 31,522.5 kilograms of corn harvested in cob and 21,300 kilograms of corn harvested in grain were quantified. In comparison with historical industry averages for yellow corn, which indicate an expected production of 7.5 tons per hectare, the production obtained represents an increase of 42% for the 2 hectares of cultivation. Additionally, a germination test was carried out on both treated plots, and it was observed that the plot treated with the technology showed a germination capacity of 100%, while the untreated plot showed 80%. It was also noted that up to 2 kilometers around the area treated with structured water, other crops benefited indirectly, including the plot not treated with technology directly. In addition to quantitative evaluation, representative samples of the produced grain were taken for physical-chemical analysis, following international AOAC 930/XXX regulations. The results of the physical-chemical analyses, as well as the toxicological analyses carried out in 2 independent laboratories, demonstrated that levels of aflatoxins and ochratoxin A were below internationally accepted quantification limits, supporting the designation of green organic food due to the restricted use of agrochemicals and the good agricultural practices implemented in the cultivation. The conclusions drawn from the trial demonstrate the significant impact of CZF technology on corn cultivation, particularly regarding irrigation water treatment. This effect was reflected in various aspects of the crop, including root integrity, environmental vaporization, and the entomological biological chain, both in plots treated directly with technology-treated irrigation and conventionally irrigated plots. The use of water treated with the technology under evaluation resulted in a quantitative increase of 42% in the total crop production. Additionally, a qualitative improvement in grain parameters was observed, with a 15 to 20% increase in soluble solids in tender cob, as well as in the protein nutritional contents of the final dry grain. These qualitative benefits of the grain suggest the need to evaluate its subsequent use as food and agro-industrial raw material. It is anticipated that the treated corn could provide better yields in animal feed science and in seed material production in future crops, thanks to the potential genomic improvement of the grain.